Allogeneic therapies are off-the-shelf treatments derived from the immune cells of healthy donors. With manufactured allogeneic therapies, many patients can be treated from the cells of a single donor. Today, the Leukopak lady takes you on a visual journey, showing you how a leukopak from a healthy donor becomes a therapy for sick patients with B-cell acute lymphoblastic leukemia. Every allogeneic therapy starts with a donor, someone who provides their immune cells to help sick patients they will never meet. Before the collection starts, the donor is consented, screened, and qualified. The collection center makes sure the donor is free of infections such as HIV, hepatitis, and syphilis, and that she is not at risk of transmitting those infections or others. Then, the donor is connected to an instrument that uses a process called leukapheresis to collect approximately 10 billion white blood cells, or leukocytes, from her blood. This process typically takes 3 to 5 hours. Once the donation is complete, the leukopak is placed into a transport container and monitored under controlled conditions. This helps preserve the quality of the cells as they're being moved from the site of collection to the laboratory for testing and processing. Once the leukopak arrives in the laboratory, a scientist removes it from the transport container and inspects the bag. The scientist completes chain of custody paperwork to document that the leukopak arrived in the laboratory in good condition. Next, a laboratory scientist takes samples from the leukopak to test for sterility, obtain cell counts, and perform characterization on the collected cells. These tests provide essential knowledge for the processing and manufacturing steps that will follow. Because leukopaks typically travel long distances to reach the manufacturing facility, they're often cryopreserved on the same day they are collected to help maintain the quality of the cells during transport. This process involves introducing a cryoprotectant into the bag, which is a specially formulated agent that protects the cells from the harsh conditions of freezing at ultra-low temperatures. In the presence of a cryoprotectant, the cells are slowly frozen to below -150 degrees Celsius. The cryopreserved leukopaks are stored in liquid nitrogen until they're ready to be shipped. When that time comes, they're loaded into secure dewars for safe transport. These dewars travel to the manufacturing facility by road and air. They are tracked by GPS, and the temperature is controlled and monitored at every step. Maintaining a constant temperature below -150 degrees is essential to preserving the quality of the cells. Once the transport plane lands, the leukopak is handed off to the CDMO, or the Contract Development and Manufacturing Organization, who completes chain of custody paperwork to maintain traceability and regulatory compliance. This entire journey from the donor screening to leukopak arrival at the CDMO is referred to as the first mile. The work that goes into getting the leukopak collected, tested, processed, and transported are essential steps in the manufacturing of an effective therapy. The first mile sets the stage for therapeutic success. At the CDMO, the transformation of the leukopak into a therapy begins. Manufacturing scientists remove the leukopak from the liquid nitrogen dewar and carefully thaw it and prepare it for manufacturing. The key immune cells that the therapy will be made from, like T cells, are isolated and exposed to a virus or other vector type that contains the genetic modification needed to turn those cells into cancer fighters inside the patient's body. The genetic modification is called a CAR, or chimeric antigen receptor, and it creates a CAR T cell. The cells are then loaded into a bioreactor, a specialized incubator that provides the right environment for the CAR T cells to become activated and expand. The bioreactor controls things like temperature, nutrients, and gas exchange, so the cells stay healthy and expand to the large numbers needed for multiple treatments. After manufacturing is complete, the engineered CAR T therapy is washed and prepared into its final form and dosage for treatment. The final therapy dose looks very different than the leukopak it started out as. It's a small bag of cells with powerful potential. The finished therapy is cryopreserved again and shipped to the hospital. The process of getting a finished therapy dose from the CDMO to the patient's bedside is called the last mile. Just like in the first mile, the therapy in the last mile of its journey to a patient must be handled carefully, temperature controlled and monitored, and tracked by GPS. This ensures the best quality therapy reaches the patient. The CAR T therapy dose is thawed at the bedside of a patient with B-cell acute lymphoblastic leukemia, and the engineered cells are infused into the patient. Those engineered CAR T cells are designed to target B cells and rid the patient of their cancer. This completes the journey of the leukopak from donor to treatment.